Here we broke down the main components of room air, or at least the components that we are interested in terms of the physiology of gas exchange. Oxygen and CO2, with the large majority that's remaining being nitrogen. And of the total pressure available in the room, each gas exerts its own individual fraction of that pressure, or part of that pressure. Partial pressure. So partial pressure is the gradient that we're looking for. And understanding this concept is important to realize why different gases can move in opposite directions. So there's a certain atmospheric pressure in this room, and you already know oxygen tends to move into the body while CO2 tends to move out. Same atmospheric pressure, different partial pressures. So we're not only adhering to overall pressure. If we were, all gases would move in the same direction. Partial pressures are what dictate the movement of individual gases. The numbers that I had earlier were off just slightly. I corrected them, but the main points are the same. About 160, about 0.3 millimeters of mercury for oxygen and CO2, respectively, sitting here in the room. Differences in partial pressure. Now, I thought it might be helpful to take this up briefly. It's a video that we've seen already, but I think it's a nice reminder about how gases move. This is exchange at the capillaries. We've seen this already, but I want to remind you that it's the partial pressures in neighboring compartments that dictates whether a gas will move or not. So let's just briefly recap the exchange or the diffusion of items at the tissue level, because this is what we're talking about today.
Substances through the capillary wall. Diffusion from the bloodstream into the interstitial fluid, or vice versa, is solely dependent upon the substance's concentration gradient.
Partial pressure gradient for gas.
Lipid-soluble substances and gases can pass directly through the thin cells of the capillary wall. Lipid-insoluble substances diffuse through pores in capillaries. Changes in diffusion gradients of nutrients, gases, and wastes may be the result of aerobic cellular respiration within each cell. Active cells consume oxygen during the production of ATP in the mitochondria. As more and more of the intercellular oxygen is used up, its concentration becomes less than that of the surrounding interstitial fluid, causing oxygen to diffuse into the cell. As the oxygen level in the interstitial fluid decreases below the concentration in the bloodstream, oxygen diffuses out of the capillaries into the interstitial fluid. Carbon dioxide is given off during ATP production. When the accumulation of carbon dioxide within the cell exceeds that of the interstitial fluid, the carbon dioxide diffuses out of the cell. As the carbon dioxide level in the interstitial fluid increases to a higher concentration than in the bloodstream, the carbon dioxide diffuses into the bloodstream.
Okay, so that's review. We've seen that before, but I wanted to reinforce and highlight the stepwise movement of gases. Now, they said concentration throughout this video. The more correct term would be partial pressure. Partial pressure of oxygen is lower in the cell, and so it moves inwards. Partial pressure of CO2 is higher in the cell, and so it moves outwards. And this phenomenon we'll refer to as internal respiration, which we'll talk about in brief before outlining the system as a whole. So we've covered the idea of Dalton's law. Every individual gas exerts a part of the total atmospheric pressure. Dalton's law dictates or defines the gradient by which the gases move. Now, whether a gas will move or not depends on where it's moving, what it's moving into, if there's room for it, if it's easy to be accepted into that space. Henry's law specifically deals with dissolving a gas in a fluid, which probably way back before we had evolved was what we attempted to do. We hadn't developed hemoglobin and red blood cells yet. And this is the idea here is trying to dissolve a gas into a fluid, plasma in the blood. If you want to dissolve a gas into a fluid, you can affect some things like temperature, but the characteristic of the fluid is predefined. And the characteristic that I'm referring to is the solubility coefficient. Just a natural characteristic of whatever fluid you're trying to dissolve gas into that's unchangeable or not very changeable. So if you have a high solubility constant in a fluid, there's a high likelihood that gas will dissolve in that fluid. If you have a low solubility constant, it's very hard to dissolve gas into that fluid. And this is a problem for us because plasma as a fluid has a very low solubility coefficient for oxygen. Plasma will not readily dissolve and contain oxygen, the gas, without some other mechanism in place. If we only had plasma, we would be hard-pressed to oxygenate the blood. It would not dissolve in. So we came up with this brilliant plan to get around that by introducing hemoglobin, which binds oxygen readily. It overcomes the idea of having to dissolve gas in the fluid. Instead, gas is attached to a molecule that happens to be traveling in the fluid. We overcome the limitation of dissolving gas in the fluid. Now, Henry's law makes the stipulation that if temperature is the same, solubility coefficient is the only driving factor in whether a gas will dissolve or not. And for us, we don't ever worry about temperature. Our body temperature stays pretty constant. It doesn't vary much more than a degree either way of 37 degrees on a daily basis. So it doesn't have a large effect. But beer makers are really happy that they can cool beer down. The reason that beer is carbonated or soda is carbonated is that CO2 is dissolved in that fluid. Admittedly, CO2 has a slightly higher solubility coefficient, so it's easier for it to dissolve in a fluid. But when you cool the fluid down, you stop all the molecules moving around and they get lazy and lackadaisical. It increases the likelihood that a gas will dissolve. The reason that we can carbonate beer and pop and soda is that we can load that fluid with a gas at a low temperature. We don't have that luxury in the body. We can't cool ourselves to encourage the loading of O2. So hemoglobin is what gets around that. Now, as we discuss gas exchange today, we're really discussing this slide shows two sides of the same coin. The book divides these into internal and external respiration, and admittedly we'll know. Well, I'm just seeing some of the animation of the next slide didn't quite work. We refer to the movement of gas from the air into the body as external respiration, and the movement of gases from the blood to the tissues as internal respiration. That's one way to separate those locations of gas exchange. But the overall process is the same. The directions of gas movement are different because the partial pressure gradients are different. So if you take one thing away from this class, individual gases move according to their partial pressure gradients. And then if you take that away from class, you don't really need to worry about internal versus external respiration. If you're given a schematic and you can see partial pressures, you know which way the gas will move from high to low partial pressure. It's also common sense, I suppose, that O2 moves in, CO2 moves out. If you're at the tissue, it's internal. If you're at the lung or the alveoli, it's quote-unquote external respiration. So external would be where the capillaries are shown here at the lungs. Internal isn't shown on this figure because we chopped off the rest of the arterial circulation. It would be down here in the cells, wherever that would happen. So let's explore the movement of gas. And we'll start with oxygen first. And we'll set the stage in a room just like this. We've already calculated partial pressures in a room just like this. O2 partial pressure is about 160 millimeters of mercury. It's high. CO2 partial pressure is very low, and I'm not going to emphasize CO2 very much for the first half of the slide. We'll come back to it as we complete the circuit. So being that partial pressure of O2 is the highest it's going to be in the room right now, when we breathe, when we ventilate, we move air in, and oxygen moves according to its to its partial pressure gradient. Inside the alveoli, the partial pressure of oxygen is low, or lower. I won't say low. So from 160 outside to just north of 100 in the alveoli, oxygen moves from the air around us into our lungs. Now I think trying to color the air within our airways in the alveoli is worthwhile. I think of it as a gradient, just a nice constant gradient from 60 in room air from 160 in room air to 105 in the alveoli. Oxygen might look something like this. Depending on how you're breathing, as it moves through the trachea, the bronchi, all the airways, down to the alveolus, might be a nice, even distribution. It might not. But generally, the concentration gradient looks something like that. So alveolar air has less PO2 or a lower PO2 than room air, and the opposite is true for CO2. CO2 is low in room air, and that gradient increases. It becomes higher as you move to the alveoli. So ventilation doesn't necessarily do much to make the gradient. The gradient is there because gas exchange occurs. Ventilation will refresh or renew or help supply new air to the alveoli for gas exchange to occur. If that makes sense. So we have an oxygen molecule in the alveoli currently. We have the alveolar sac, individual alveoli shown here as a cluster of grapes, and we need to see where it will move next. Through a process of simple diffusion, it moves from the air into the blood. The membrane is very thin, the surface area is very large, and there is a partial pressure gradient. But it's not very big. Partial pressure gradient in the arterial blood is only a little bit less than the partial pressure of oxygen in the alveoli. So the main reasons that oxygen moves with this low gradient are because the surface area is large and the membrane is thin. Now once oxygen has been loaded into the blood, all arterial blood is the same. It's distributed throughout the body, returned to the heart through the pulmonary veins, pumped through the vasculature to the tissues that use it. And you can see the partial pressure shown here. 100 millimeters of mercury in the arterial blood compared to 105 in the alveoli for O2. So it still follows the gradient, but it's not a large gradient. The gradient becomes a little bit larger as we move into the cell. Now, depending on which cells you're referring to, these values can change. These values are representative of a normal cell with an active basal metabolic rate, a liver cell, a brain cell, a muscle cell. These cells are always active, always using oxygen. So their PO2, their partial pressure of oxygen, is lower. Here it's 40. Well, if we knew nothing else except the partial pressure in the arterial blood and the partial pressure of oxygen in the cell, we'd know that oxygen would move in, down the partial pressure gradient. Now, what that video alluded to that's not shown here is where the oxygen goes once it's in the cell. It's used for something, and we talked about this last semester. Aerobic respiration or oxidative phosphorylation uses oxygen to make energy. It does that by making water. All the oxygen you breathe in ends up as water in the body. The reactions behind that we won't talk about in depth. You can ask your biochem prof or your metabolism prof when you get there, but all the oxygen that you take in ends up as water. If the average PO2 in the cell is about 40, the site where this conversion to water happens, the site that consumes oxygen, has a PO2 of about 5. PO2 of about 5. And I say about because it's on such a small scale that we require such sensitive instruments that we can hardly measure it. But it's about 5. It's as close to 0 as we care to approximate. And so you'll notice through this whole process, the gradient dictates movement. From room air to the alveoli, a little hop into the blood to the cells, and then down to the mitochondria where the PO2 is lowest. We see this progressive decrease in partial pressure, pushing oxygen further and further into the body where it needs to go. Oxygen has no choice but to follow this gradient. It can't move against its partial pressure gradient. So understanding this concept already sets you up to understand the second half, which is CO2 production. Gases move according to their partial pressure gradients. Where does CO2 come from? CO2 actually comes from the breakdown of food. As you process food in metabolic pathways, part of those pathways spin off CO2. It's actually a separate process than the one that consumes oxygen, but they're very closely related. So food is what produces CO2, or specifically the breakdown of food. That happens somewhere in the cell, and it spins off CO2. The more food you consume, the more CO2 is produced. At the level of the cell, as CO2 accumulates, we have a slightly higher partial pressure of CO2, 45 millimeters of mercury, that you can see. Actually, what I'm realizing now is this isn't as clear-cut as the oxygen side. The PCO2, the partial pressure of CO2, here is listed as the same in the cells as it is in the venous blood. Now, let me ask you, for your body to expel CO2. Are these going to be equal? Is it possible that PCO2 is equal on the venous side, in the venous blood, and in the cells? If you're going to exhale CO2, is that possible? If you want CO2 to move from inside a cell to the lung, what does it have to follow with? Along the what gradient? I heard some murmurs. Partial pressure is right. Good. Is there a gradient here? As laid out on this slide, with PCO2s of 45 on the venous side and in the cells, is there a gradient?
No.
No, thank you very much. Appreciate it. No, there's not. There's no gradient. So at least according to this schematic, there would be no movement of gas. We know we exhale CO2, and so what I'm trying to get across is that this is not possible. Maybe it's a small difference, but we would accumulate CO2 that would bleed out into the venous blood. We know we expel CO2. There has to be a gradient. Maybe it's imperceptibly small, but it's there. It's not shown on the slide. If 45 and 45 were the true numbers, there would be no movement. But of course there's movement. This is not the case. Anyways, that's a tangent. Let's get back to the movement of CO2. There's some gradient that pushes it into the venous blood, even though it's not shown here. And as the venous blood moves, mixes in the right heart, sent back to the lung to be oxygenated and to lose CO2, the partial pressure drops. There is a gradient in the alveoli from 45 to 40, encouraging CO2 to move out. And there is a large gradient from the alveoli to room air, encouraging CO2 to be exhaled or dispersed with the breath, with normal breathing. Like the O2 on the arterial side, that follows its cascade of partial pressures, CO2 does the same on the venous side. And if you're going to take anything from this class, individual gases move according to their partial pressure gradients. I want to bring up two other things before we leave this slide that I just thought about. So they're not written on here, but two things. All arterial blood is the same. It's mixed. It's homogeneous. Not all venous blood is the same. And I've said this before, so you might know this already. Not all venous blood will have the same PO2 and PCO2. If you take a liver cell and a muscle cell sitting here at rest, maybe they do. Maybe they have similar partial pressures of oxygen and CO2. But you're not content sitting here. You jump on an exercise bike and you start to ride to get a good workout in. Your muscle is active, your liver's not. Or your muscle is more active and your liver is less active. Your muscle would produce more CO2 and it would consume more oxygen. So for venous blood draining a muscle, PCO2 would be higher, PO2 would be lower. All that means is that there is a greater likelihood for gases to move in more metabolically active tissues. The gradients are bigger. Those were the two things. One, not all venous blood is the same. Two, the gradient changes with the activity, the metabolic activity of that cell. The numbers that are shown here are the average venous blood, taking into account all the cells in the body, if you were to mix them all. Is anything about this concept not clear before we move on? Okay, good. because we're going to take a look at how oxygen and CO2 are carried. This shows us that they move. There are partial pressure gradients, but how are those gradients achieved? How does the blood carry these gases? Oxygen almost exclusively is carried by hemoglobin. Remember, according to Henry's law, the solubility coefficient dictates whether a gas can be dissolved in a fluid. Plasma has a very low solubility coefficient for oxygen. 1.5% of the oxygen that you breathe and transport is dissolved. That's not very much. In contrast, 98.5% is carried by hemoglobin. The vast majority is carried by hemoglobin. If we didn't have red blood cells, if we didn't have hemoglobin, you could see we'd be in a pickle. Very little ability to carry and transport oxygen. CO2, on the other hand, isn't as limited. Plasma has a higher solubility coefficient for CO2. So more CO2 dissolves in the plasma, 7% as opposed to 1.5%. But still not very much. It's more than oxygen, but it's still not very much. Some of it takes the place of oxygen and binds to hemoglobin. But hemoglobin isn't designed to bind CO2. That's not its purpose. It can fit. And some of it will tag along with hemoglobin on the venous side. But that's not what it's designed for. It's purpose-built to bind oxygen. The majority of CO2 is transported in the blood as bicarbonate. Bicarbonate is this ion, HCO3. There should be a negative symbol there because it has a slightly negative charge. HCO3 negative is a bicarbonate ion, and this is produced by that enzyme carbonic anhydrase that I mentioned last class. We'll see the reaction later. CO2 is taken up. The enzyme carbonic anhydrase makes bicarbonate. The bicarbonate dissolves easily in the blood and is carried through the venous system to the lungs. 70%, almost 3/4 of CO2 is transported as bicarbonate. And so in this slide that you see in your series of notes, same exact principle. We have external respiration at the lung, internal respiration at the tissues. We have the arterial system shown here on the left, but that is diffused throughout the body, and the venous circulation on the right-hand side. Now we're looking at the fractions or the percentages of oxygen and CO2 and where they're carried, not partial pressures. You can see the 1.5 and 98.5% of oxygen on the arterial side here, and CO2 shown on the left. By far, hemoglobin carries the majority of oxygen. And if you were to measure it, if you took out 100 milliliters of your blood, put it through an analyzer, your arterial blood would contain 20 milliliters of oxygen. Every 100 milliliters of blood has 20 milliliters of oxygen. Every liter has the equivalent of a juice box of oxygen. Of that 20 milliliters, 19.7 is bound to hemoglobin. The large majority is bound to hemoglobin. Then, like we saw on the venous side, hemoglobin can bind oxygen. It doesn't do it as well as it binds oxygen proper. The majority is transported as bicarbonate. So in this next section, what we want to look at are the mechanics of gas transport. The mechanics of O2 binding to hemoglobin and the mechanics of CO2 being transported in the blood. We'll look both at binding to hemoglobin, but primarily we're going to look at how it makes bicarbonate and how that, well, once it makes bicarbonate, it just travels along in the blood, but how it reverses when we need to expel CO2 from the body. So we're looking at those mechanics in this next section. We're going to start with oxygen and hemoglobin. That's probably, it's not most important, but that's the signature gas in gas exchange. And we'll move to CO2 next. Now the transport of oxygen by hemoglobin is defined by the HbO2 association curve. This describes how well hemoglobin binds oxygen. And it looks like this. The HbO2 association curve. The first thing you're going to notice, well, let's take a look at the graph first. We have, for a given partial pressure of oxygen, we know that term really well, how saturated is hemoglobin. And saturated is a word that we use to describe how much oxygen is it holding. If all four heme sites within the hemoglobin, and all the hemoglobin molecules are fully bound with oxygen, it's fully saturated. If some of the sites have oxygen, some of them don't, it's partially saturated. And this y-axis, the scale, tells you what percentage of all of the sites are bound. If every hemoglobin molecule has 3 sites bound with oxygen, one that's not bound with oxygen, the total blood saturation is 75%. Okay, so that's the graph that we're looking at here and over the next few slides. What you'll notice after understanding that is, for a high PO2, hemoglobin loads oxygen. If you have a high partial pressure of oxygen, hemoglobin will carry it. If you have a low partial pressure of oxygen, less hemoglobin is saturated. Now normally, to describe that kind of relationship, that proportional relationship, if one goes up, the other goes up, you'd expect it to look like a straight line. As PO2 went up, saturation went up. Normally when we see these kind of proportional changes, we're used to one linear line that describes the relationship. This is obviously not linear. And I'm going to wait on telling you why that is. It's obviously not linear, and it has to do with just a natural characteristic of hemoglobin. We'll talk about that in a second. Let's hit some landmarks that we know. PO2 in room air, 160. PO2 in the alveoli, 105. PO2 in the arterial blood, 100. At a PO2 of 100, hemoglobin is essentially completely saturated. All of the sites are bound. It's carrying as much oxygen as possible, which is good, because this situation occurs at the lung. You want the blood to be loaded with oxygen, you want it to be oxygenated, you want hemoglobin to be fully saturated. And at the PO2 that we encounter at the alveoli and on the arterial side, luckily hemoglobin is fully saturated. Now this doesn't change as you move through the systemic circulation down the brachial artery to the arm and the fingertips, or through the femoral artery to the leg. It stays fully saturated. Only when you get to the tissue and PO2 is lower does the saturation get affected. Now let's say that we get to a liver cell or a muscle cell. Remember what PO2 at those active tissues were? Ballpark? Can you read the figure? It's on here. Blood in the systemic veins is approximately what we're looking at the cells. As we reach the active cells, PO2 drops to about 40 millimeters of mercury. Even then, hemoglobin's still 75% saturated. 3 of every 4 sites still binds oxygen. Maybe that's surprising. Maybe you thought all of the oxygen that you brought in and loaded in the blood was delivered to the tissue. That's not true. Only about a quarter of the oxygen that you load into the blood gets used by the tissue at rest. Which is actually really clever if you think about it. Because what happens when that tissue becomes more active? If we used all of it, it'd be SOL. Since we have some oxygen left over, we have the capacity to extract more oxygen from the blood if we are at a tissue that's very active, that's very metabolically active. And you might see this other dashed line shown on the graph is meant to represent a muscle cell during heavy exercise. Very metabolically active tissue. PO2 is very low because the mitochondria are using a lot of oxygen. As the PO2 drops to 20, in this case, the blood that leaves that muscle is only 35% saturated. What I'm describing is the ability to extract oxygen. What just happened here, the muscle became more active and it increased its extraction of oxygen. You'll hear that term thrown around a lot. So normally at rest, venous blood, still 75% saturated. Hemoglobin is 75% saturated. If you move to exercise, the blood draining those muscles that are very active, 35% saturated, still not zero. And in fact, you'll probably never encounter a situation where it gets all the way to zero. Maybe intense exercise on a mountain peak, you're probably more likely to pass out before you can measure hemoglobin saturation close to zero. What else do we notice about this curve? What's really nice about the shape of this curve is that hemoglobin is fully saturated if PO2 is at least 60. Essentially fully saturated. So a PO2 of 60, if I trace that line all the way up, I'm on this upper flat portion of the curve, and maybe I'm 95% saturated. For all intents and purposes, that's pretty saturated. This is one of the reasons why we're thankful there's not a linear relationship. If this were linear, at a PO2 of 60, I would have a much lower saturation. I would be a lot less likely to tolerate moving from Nova Scotia at sea level to what's nearby that's tall. There isn't much. We're all pretty much at sea level here. But let's say you flew to Colorado. You're in Denver for a while, 5,000 feet up in the air. You would not tolerate that really well if this were a linear relationship because hemoglobin would be far less saturated. This characteristic allows us to move to altitude where PO2 is lower and not be too bad off. So hemoglobin fully saturated if we are at least at 60 millimeters of mercury. If you're interested, you could back calculate what atmospheric pressure that would be, knowing that the percent of O2 is the same. I'm not going to do that here. And I wanted to point out to you that at rest, hemoglobin's still at least 75% saturated. You only extract 25% of the oxygen in a resting tissue, which might be, what is that, 5 to 6 milliliters of oxygen per 100 mils of blood? Not very much. Now, we're not going to get into these other factors in depth. We'll maybe trace out one or two of them. But this curve moves. Actually, maybe what I'll do quickly, I didn't write this on the slide, but I'll tell you why it's shaped this way. Why it's shaped this way. Hemoglobin, with nothing bound to it. You have 4 sites that can take on oxygen, right? When all those sites are empty, it's actually difficult to bind oxygen. If you bind one site with oxygen, there's a change in the shape of hemoglobin. And that change in the shape makes it easier for the next site to bind oxygen. And if 2 are bound, it makes it easier for the third to bind oxygen. So what I'm describing is this acceleration that you can see here. It's hard to load oxygen initially, and it might even be a bit more flattened in practice. But as you bind oxygen to more sites on the hemoglobin molecule, it gets progressively easier and easier. So we get this rapid binding of oxygen to a point where everything is almost full. And it's a little bit like playing musical chairs and only having one chair left. If there's a lot of you trying to get to that one chair, it's hard to bind that last space. When the hemoglobin molecules are mostly bound, it's difficult for oxygen molecules to find a place to bind. They're mostly full. And that describes this leveling out of the curve at really high PO2s. They're mostly full. It's hard to find a place to bind. So as you bind one, it gets progressively easier. And then when most of them are bound, it's hard to find a spot for the O2 to load. The shape overall we call sigmoidal, or S-shaped. This is distinct. It's often used to refer to hemoglobin specifically. It's a very unique shape in physiology. So class is done. We will come back next week and recap how these factors affect the shape of the curve. It turns out that some of these factors are tailor-made to help us load and unload oxygen with exercise. There might be some questions about these factors in the assignment or the activity that I open up for you tomorrow, and that will stay open Saturday, so keep that in mind. Have a wonderful break. See you in a week and a half. It's going to be a long time.